Good morning, everyone. Welcome to the Yale Community for New Haven, or the Yale Office of New Haven Affairs Community Breakfast. We host these community breakfasts every month during the academic year, and we are happy that you are joining us this month. My name is Karen King. I am with the Office of New Haven Affairs. I will add a link in the chat box so that you can add your name to our mailing list so that you can continue to be informed about our upcoming events every month. Before we get started, I'd like to make a few announcements. The first is, and I'll share my screen here, the Yale, the future leaders of Yale and the Yale African American Affinity Group are inviting people to join a panel discussion designed for young women of color between the ages of 9 and 18 that will focus on the importance of empowerment, positivity, and overcoming adversity. Attendees will hear inspirational stories from Yale leaders and students, participate in a Mindful Monday empowerment exercise, learn about Yale University as an attainable option for education and employment, and participate in an environment where girls can talk freely and be themselves. This event will be held on Monday at noon. I will put the um, registration link in the chat so that you can share and or join. This event is going to be hosted by Fallon Thomas of the New Haven Hiring Initiative. And speaking of the New Haven Hiring Initiative, the Yale New Haven Hiring Initiative is an office that is focused on hiring New Haven residents for jobs here at Yale, and particularly from neighborhoods of focus. Fallon Thomas is part of a great team that is going around to different neighborhoods by Zoom now to get the word out about Yale's proprietary application system called STARS, how to create a profile, how to upload your resume, upload your cover letter, and do a job search and submit to be considered. So I would encourage you all to visit their website. I will put this in the description as well, so that you can stay up to date on their events, their upcoming events. But again, it was a great presentation last night. Last month, if you joined us, you had the pleasure of hearing from Risa Nelson, Dean of Yale College and the director of the Afro-American Cultural Center. She wants to remind everyone that while the Afro-American Cultural Center celebrates Black History all year, they do have events that are for Black History Month. I will put their website in the description so that you can visit and learn more about what's going on, not only this month, but all year. I also want to remind everyone about the Yale Community for New Haven Fund. This fund was set up by Yale University in April, and it was designed to provide funding to New Haven organizations, nonprofits, as they're supporting residents here in New Haven who've been negatively impacted by the pandemic. The focus is to provide funding to provide basic needs for New Haven residents. So food, PPE, good mental and physical health, bridging the technological divide that exists for some New Haven public school students as they're trying to access their studies remotely. I want to again encourage local organizations to apply for funding. There is still funding available. We have a particular interest in finding first-time applicants because we realize there are plenty of organizations out there who are doing great work, and we want to during this pandemic, and we want to try to support their COVID-19 related work as they're supporting New Haven residents. I will put the link to this website in the chat as well. There is an application here for nonprofits to apply, and nonprofits can take a look at the nearly 200 organizations that we've supported with nearly $3 million in funding. And again, there is still funding available. On to our featured speaker. Joining us today is Dr. Saad Omer. He's the inaugural director of the Yale Institute for Global Health. He's also professor of infectious diseases at the Yale School of Medicine and the professor of epidemiology of microbial diseases at the Yale School of Public Health. Dr. Omer has conducted studies in countries around the world, and his research portfolio includes epidemiology as respiratory disease viruses such as influenza, RSV, and more recently, COVID-19, clinical trials to estimate efficacy of maternal and/or infant influenza, pertussis, polio, measles, and pneumococcal vaccines, and trials to evaluate drug regimens to reduce mother-to-child transmission of HIV. Moreover, he has conducted several studies on interventions to increase immunization coverage and acceptance. His work has also included public health preparedness strategies to effectively respond to large emerging and re-emerging infectious disease outbreaks. Dr. Omer's work has been cited in global and country-specific policy recommendations and has informed clinical practice and health legislation in several countries. Dr. Omer has published widely in peer-reviewed journals, including the New England Journal of Medicine, JAMA, Lancet, Pediatrics, American Journal of Public Health. Moreover, he has written op-eds for publications such as the New York Times, Politico, and the Washington Post. Dr. Omer has received multiple awards, including the Maurice Hillman Award by the National Foundation of Infectious Diseases for his work on the impact of maternal influenza immunization on respiratory illness in infants younger than 6 months for whom there is no vaccine. He has served on several advisory panels, including the U.S. National Vaccine Advisory Committee, the Presidential Advisory Committee on Combating Antibiotic-Resistant Bacteria, and the Vaccine Innovation Working Group, and the WHO Expert Advisory Group for Healthcare Worker Vaccination. Moreover, he served as an academic affiliate of the Office of Evaluation Services, formerly known as the White House Social and Behavioral Sciences Team. Please help me welcome Dr. Saad Omer.
Thank you. It's my pleasure to be here. And I'll be talking about vaccines, and I'll use a few slides initially, and hoping to have some time for questions and answers, because I know around COVID-19 vaccines, folks have several questions that they usually want to discuss. So I have tried to set aside some time for questions and discussion, etc. So if I can share my screen for the overview. I'm assuming everyone can see my screen, correct?
Yes, we can all see it.
Okay. All right. So this has been the progress in terms of vaccine development around COVID-19 has been a bit of a miracle. Within a year, we had 4 approved or authorized vaccines. Actually, now it has grown to longer. 5, 6 are either approved or authorized. The difference between authorization is usually emergency use authorization. That's also goes through really rigorous review, but it's not quite the full licensure because the regulatory agencies retain the right to revoke that. And so on top of that, the biggest success we have is having a robust portfolio of vaccines in the phase 3 trials. So the more we hedge our bets, the better it is for all of us. In terms of the more candidates we have, the better it is. And just to take you through how this was achieved quickly, and because people have a lot of questions about the process, how it unfolded over the last year. And just to tell you that the process, while against this virus started early last year, it didn't truly begin just on that date. It actually began much earlier when we started doing 2 things: investing in the so-called platform technologies, and a concept specifically for pandemics that was developed was of platform technologies. And what do I mean by platform technologies? Meaning that even when you don't know what disease will emerge, you have an idea of what kinds of families of viruses, et cetera, pathogens, these kinds of large-scale threats can come from. So therefore, you invest in technologies that can be quickly modified to respond to that kind of an outbreak. And in that case, for example, the technology which saw a lot of investment was intellectual investment mostly, but some resources as well, was the mRNA technology that people, I'm sure, have heard about it by now. And the viral vector technology, where you take one virus that doesn't cause disease in the human host and put a protein on top of it, which then becomes your vaccine. Put a protein of that you want to target eventually for your immune response on top of it. So you create these viral vectors. So all of these technologies took a lot of investment even before the pandemic. But on top of that, there has been some innovation in thinking in terms of regulation, etc., around uh, um, uh, there has been some innovation in thinking um, around how we evaluate these vaccines and how to do that quickly in a public health emergency. And one innovation, and that was implemented here, is to figure out how to do trials. So what we do is we identify a target on the virus, which should be the aim for our vaccine, with on which we should target our immune response, and that is likely to be successful. Then often research entities partner with biotech companies or pharmaceuticals to sort of share sort of that knowledge and co-develop the vaccine itself. And sometimes pharmaceutical companies have their own technology in-house. But then the idea is that you do initial, what we call pre-clinical work, the work that, and especially the work that is essential before you move to human trials, fairly quickly. And then the rest of it that doesn't require, that doesn't need to be done before human trials, in parallel with human trials. And as you go into human trials, what you do is you eliminate the dead space between various phases. You must have heard of phase 1, 2, 3. In phase 1, we vaccinate small number of people, and we to look at initial safety and immune responses, not efficacy, meaning not efficacy against what we call clinical endpoint with against actual disease, but immune responses initially. But then we expand the size with each successive phase and go from phase 1 to 3 trials, where in phase 3 trial we look at actual disease related outcomes as well. In this situation, what happened and what we will be doing increasingly around pandemics is to combine those phases, recruit the same number of people, but quickly, rather than doing one phase, stopping, and then deciding whether it's worth going forward or not. What you do is you look at the data on an ongoing basis, and then you enroll the next set of patients right away without waiting, without that sort of that regulatory dead space. The other thing, what was done is, so if you think about, if you want to do a large trial, um, where you need 30,000 people, so you can have 10 sites with 1,000 people recruitment target each. As you can imagine, it'll take some time to recruit those people, even if the follow-up is the same. But if you want to increase the number of, if you want to do it quickly, what you could do is, 10 sites instead of 10 sites with 3,000 people each, sorry, you can do 30 sites with 1,000 people, or you could do 60 sites with 500 people, or you could do 120 sites with 250 people, or 240 sites with 125 people each. So when you increase the number of recruitment sites, and this country has the capacity to do that, fortunately, what you do is you recruit in each site, that you achieve the target in each site more quickly without compromising the amount of follow-up you would be doing, because your recruitment happened quickly. So that's, just to say, it wasn't all sort of going to 240 sites, but the number of sites was much, much higher than the usual trial sites, because we had resources to do this, and we went in as a country and as a world directly to recruiting a lot of people. And the other thing, what happened differently, is that a lot of manufacturing and scale up that happens after licensure or after authorization happened in parallel. In the future, frankly, I think we should do even more of what we call at-risk manufacturing, uh, so that the shortages don't happen. But I think even now it was sensible to do a lot of parallel manufacturing. So I just wanted to take some time to explain to people what happened. How did we get from a target identified in January of 2020 to an authorized vaccine in this country in December 2020. So let me go through a little bit of overall virology and what vaccine platforms, when you hear about different vaccination strategies, what do we talk about? I will focus on a few select of categories of vaccines, but here it is. This is the virus. This is how actually all, pictorially and overall, all coronaviruses look like. But within them, there are differences, obviously, at a more micro level. But essentially, what you have in SARS-CoV-2 is the so-called spike protein, which protrudes, and that it lets it attach and enters different cells. So, as you know, viruses attack our own cells, and that's why they're so um, dangerous to us, or that's why they can be very effective. The other thing, kind of protein that we have is the so-called matrix protein that keeps the integrity of the virus together. There's the so-called envelope protein with similar functions without going into too much detail. And then you have the nucleoprotein, which has the viral RNA, etc., as well. So we use different for different kinds of purposes. Some are used uh, as targets for testing. But for vaccines, it was identified earlier on that the so-called spike protein, and we knew that before, we knew that, or we had a good idea even before this virus emerged, based on our experience with SARS-1, which was like a couple of decades ago, and MERS, the Middle East Respiratory Syndrome virus. And so these were other coronaviruses. So we knew that the spike protein will be the most appealing target for this. So most, but not all of these vaccines that are there actually target the so-called spike protein. So this is a blow up of this that kind of looks at a more micro level. And this protein has the so-called receptor binding domain, which is the even a further target where it latches onto the cells, our own cells, etc. So how do we target this? First of all, is a broad-based one where we create a vaccine, and which is not being used in the U.S., but actually I may discuss about that as an international vaccine that you may hear about it. You take the whole virus and inactivate that so that the disease-causing potential goes away. The other one that is being considered, some of these are theoretical vaccines or early stage vaccines that didn't move forward to advanced stages, so I'll skip over those. The other one that is there is a recombinant spike protein. So they create the spike protein directly through a process, and that spike protein is given directly. I would say that then the two more important one are these two replication competent and replication incompetent vector viruses, where you take another virus where you that does not have the potential to cause disease in humans. So rather take a chimpanzee virus. So the virus is a virus. It's not a chimpanzee DNA. But it causes disease in virus. It causes diseases in chimpanzee or other human viruses that don't cause severe disease. You take that, insert the spike protein onto that to create a kind of a chimera, and that is used as a viral vectored vaccine. And some of them can replicate in the body to have a different kind of response. Others do not replicate. And then the other important one is the mRNA vaccine, which is the messenger RNA, which and covered with the so-called lipid nanoparticles. So like fat nanoparticles, tiny particles that coat the mRNA so that it can actually survive in your body and target the cells where we want to go. So that's the overview. So how does an RNA vaccine work? So ignore this part. We're not initially, there were DNA vaccines in development, but we're not, those are not the frontline vaccines anymore. We are dealing with the RNA vaccines. So usually, even without a vaccine, what happens is that you have this nucleus that has our own cells that have the ability, um, or what the way they cells function is through development of proteins and production of proteins, etc., for various purposes. What happens is that that message that of the code for that protein is in our own DNA, but the machinery to produce these proteins is outside in our cells. So what happens is that our cells send the signal from our genetic material uh, to the protein manufacturing machinery of our body. There's entities, for example, called ribosomes that create these proteins through the so-called messenger RNA. So it's essentially like an email or I often think of a Snapchat message that goes in and then gets destroyed. The message itself gets deleted. Think of it as a Mission Impossible auto destruct message that you send a message, it does its job and then goes away. So that's what messenger RNA is. They're intentionally, in a sense, that by nature are highly unstable molecules in the sense that you don't want them to hang around and keep on giving the message. It comes from the nucleus, it goes to the protein manufacturing machinery of your cell, which is outside the nucleus. So it's a kind of a one-way process there. And then you create the proteins, and proteins do whatever that you want them to do. In the case of RNA vaccines, you stabilize the messenger RNA that you create, that a vaccine manufacturer or a sort of a scientist create, package in a lipid nanoparticle. So it's important to package it in a lipid nanoparticle because, as I said, that mRNA is highly unstable. It wouldn't survive. And then one of the technological advances came how to stabilize that. And it was through these different approaches, including a lipid nanoparticle. You inject in the body, this whole particle and mRNA goes to your cell, and this lipid nanoparticle facilitates entry into the cell, and then this particle goes away. But then your mRNA goes in, and then now that it's without the lipid nanoparticle, it can just go directly to your ribosomes or other, your protein manufacturing machinery. And the protein they create is like the spike protein of your own virus, of a COVID-19 virus. So the important thing is it doesn't uh, uh, interact directly with your DNA. It doesn't go to your nucleus. It's all happening outside. So some people have questions about, is it going to do anything to my genetic material? No, because it's not interacting with it. It's going to the direct protein manufacturing part of your body and creating these. So now what happens is when you have these proteins created for the first time, when your body sees it, the parts of the immune systems see that protein, they say, okay, this is an outside thing. I need to mount an initial immune response to it. And then what it does is that after that initial immune response, it remembers, the body remembers that it has seen these kinds of proteins before. So when then there is an attack of the virus itself, it remembers, it looks at these proteins and say, okay, this is something I need to mount an immune response to, and it responds very quickly to that. And so it is that kind of an approach, and this is how we get immune responses through mRNA vaccines. The viral vector vaccine, as I said, is that you take a virus that doesn't cause disease in humans or causes sort of mild symptoms in humans, stick the spike protein on top of it to create these chimeras, and it can be either what we call replicating or non-replicating virus. But the end of the story is that your immune system sees that protein, in this case, presented on top of another virus, and says, okay, I need to mount an immune response and then remember it for the next time. And that's how these vaccines work. And the reason why I'm discussing these in a little bit more details is that these are more frontline vaccines. There are a ton of vaccines manufactured by sort of different companies, et cetera. Some, the ones that are more relevant to the US population are Moderna, NIH, Pfizer, BioNTech vaccine, and AstraZeneca, Oxford vaccine. There's another one, the J&J vaccine and Novavax vaccine, that are not here, but those are also frontline for the US. The other ones are being evaluated or now even being used outside the US. So a few details about the so-called Pfizer BioNTech vaccine. It is a modified RNA vaccine that has been authorized or approved in several countries, including the US, and they went through large phase 1, 2, 3 trials. Uh, it's 95% efficacious. So what do you mean by efficacy? Efficacy essentially means that the likelihood, the incidence of disease in those who are vaccinated versus unvaccinated. So if you are vaccinated, your likelihood of getting the disease goes down by 95% after vaccination. But remember, these vaccines were evaluated against lab confirmed disease and any kind of disease, including mild to moderate disease. When you look at the data in more detail against severe disease, the vaccines seem to work really, really well, even more than 95%. So I would say 99 to 100%. So that's really good news. And these vaccines are stored. One of the things that they come with an asterisk in terms of storage is that their storage is at minus 70 degree Celsius, which is a sort of what we call ultra cold chain, which is really cold temperatures, and it's a special kind of refrigeration is needed. And then it requires 2 doses. Similarly, the next one, so this was given in an EUA. Moderna vaccine is 94% efficacious. By the way, functionally, there's no difference between 94 and 95%. They're equally effective. But the difference about the Moderna vaccine is that can be stored at minus 20 degrees Celsius. So you can imagine you can extend the vaccination uh, campaign to sort of communities, go outside, engage populations that are hard to reach or are not coming to your hospital for various reasons, including, for example, the elderly, etc. And so that is a very appealing characteristic from the immunization systems perspective. From the patient's perspective, it doesn't make a difference between these two because they're all equally effective. So that's another question that comes up. This also requires 2 doses for the full immune response. A little bit about the data. In this pandemic, CDC and FDA have stood up. There are existing vaccine safety systems that are in place. In this pandemic, they have supercharged these systems, and there's a new system in addition to the existing systems called vSafe, which is supposed to be, text messaging check-ins from CDC. You activate that, or vaccine recipients activate that, and everyone can do that. And you report clinically important events directly, and it's a semi-automated systems. And then there's another system that sits on top of it, the vaccine event reporting system. If there is something that the system picks up that needs to be followed up, then that parallel system of what we call VAERS system follows up. So based on these data, in terms of vSafe data, what we find is, uh, the early reports were, because Pfizer started early, so there wasn't a lot of data for Moderna dose 2. What it tells you is that early data suggested that was in line with our vaccine trial experience as well, that these vaccines, are hurt a little in terms of pain and fatigue, etc. But these are expected, side effects. They go away after a day or two. But it's to keep in mind that when your immune system is working, you get some symptoms like pain, fatigue, headache, etc., and then they go away. And has been the experience of sort of a lot of our colleagues here as well, that they get a little bit of fatigue, pain, sometimes chills, etc. But the pain is more of a feature. And the reason why that happens is that when our immune response responds effectively and vigorously, some of these symptoms get produced. But then the question comes in for those who, those who don't get those symptoms, and people often say, like, started asking questions that, look, I didn't get any symptoms, and then the response is no. That's great. If you didn't, that doesn't mean that your immune response is not working. But what happens is that it's a range of the type of immune response that people mount, and within that range, it's safe. But for some people there are symptoms, others there isn't. But everyone, we know from really high quality data around these vaccines that people's immune system is responding. So VAERS is the Vaccine Advanced Reporting System. This is managed by FDA. This is not text-based where you detailed reports often by physicians are filed. And this gives you another initial layer of data. There's other more sophisticated systems where people are actively followed up. That's called um, VSD, Vaccine Safety Data Link, and then there's a different similar project. Vaccine Safety Data Link is managed by CDC, but there's a different version of it that is managed by of a database, not VSD, by FDA. Based on that, and based on based on VAERS, etc., the estimated anaphylaxis, which is a severe allergic reaction following COVID-19 vaccines, is a real adverse event. It is a real side effect, and it is considered not a mild side effect. Uh, so therefore, it needs to be managed. So CDC calculated the rate of anaphylaxis, and it is, while it's real, it's rare, fortunately, and the reporting rate is 5 per million doses administered for the Pfizer-BioNTech vaccine, and approximately 3 per million doses administered for the Moderna vaccine. So qualitatively, they're similar there. There are data from UK, etc., that supports that. Fortunately, in all of these cases, while it does cause concern, uh, we have been able to manage all of these cases through um, uh, quick treatment. The implication it has is that CDC cautions those who have a history of, in almost of all of these cases, it was for people with a history of severe allergies to begin with, specifically to products that are in the vaccine. So you can assess, the providers can assess those kinds of allergies, and that's one of the reasons some people with people who have history of allergy to any vaccine component at this point, they're not getting the vaccine, but have other severe allergies, often are asked to sit for 30 minutes of observation, and everyone else is being observed for 15 minutes after vaccination. So it is, again, when you have a robust safety surveillance system, you do find these kinds of events, but these have been managed fairly well without long-term consequences of vaccine recipients. And this has been looked at in the context of, so I serve on the World Health Organization Committee, a working group that sort of helps draft recommendations for what is called WHO SAGE. But you know so we have looked at it on our side on this as well, but in the US, the CDC has looked at it, and the benefit of vaccination, even in the context of these rare side effects, heavily high, overweigh the risks, and we can manage these risks. The next one is the coming down the pike. One of the next ones is the so-called Oxford AstraZeneca vaccine. It has approximately a pooled efficacy of 70%. They did multiple trials and then combined the data from that. These trials were initially done outside the US. This US large trial is awaited. And the refrigeration requirements is very attractive. And one of the reasons people continue to look at this vaccine, and this will be the predominant vaccine in Africa, at least in this stage. It was developed by, as I said, Oxford University, and then partnered with AstraZeneca, a vaccine company, to scale it up. It's what we call a non-replicating viral vector. So in this case, they take a chimpanzee virus and insert the human, this SARS-CoV-2 spike protein, and that creates the vaccine. The attractive part is that it can be kept at refrigeration temperatures. As you can imagine, it increases access and our range of deployability of this vaccine in this country and other countries a lot. It also, at this point, is recommended to receive 2 doses. By the way, there are trials going on with mix and match, and 1 versus 2 dose trials will be starting. But at this point, the evidence highly suggests that everyone receives 2 doses. Um, then Janssen, or Johnson and Johnson, is also a non-replicating human virus that is it's a vectored vaccine. As I said, it has had initial data on phase 3 trials, and they are under considerations for another authorization in the US. Their efficacy is 72%. One thing to remember, This is efficacy against the primary endpoint of any disease, any lab confirmed disease. The efficacy against severe endpoints seems to be similar between the types of vaccines that have had detailed data being presented. Uh, the other, similar to um, uh, the AstraZeneca vaccine, that this is refrigerated at the 2 to 8 degrees Celsius, which is the normal refrigeration temperature. But this is for 3 months. Before that, you have to freeze it. But 3 months is actually all you need in this country to actually increase the range. The other thing is that these were tested for one dose specifically. They have a two-dose trial that will have results a little bit later. But one dose, if you're getting really good efficacy with one dose, especially against severe disease, this is a good, attractive option. So I'm glad that in this country we hedged our bets on different products, and so therefore it gives policymakers and public health folks options. Uh, then I will just quickly go through several other ones. This is a Russian vaccine that is being used in all sorts of other countries, but is not targeted for the US. But you may hear about it. This has temperature requirements similar to the Moderna vaccine, meaning that it's freezer but not ultra cold chain, in the sense that it's not being stored at -70 or -80. It has 92% efficacy. This is also a non-replicating viral vector vaccine. It's two different kinds of viruses. So the first dose is another kind of a viral vector, and the second dose is a different kind of a viral vector. And they did a mix and match strategy, which I think was a smart strategy, etc. Because you don't want your body to sort of you want your body to see the virus, quote unquote, very well each time. So one of the concerns was that if you see it repeatedly, the vector itself, the host virus itself, that you may not mount as good a response. So that's why they did a mix and match strategy, and that seems to have paid off. And then there is a Sinovac vaccine, which is a whole virus inactivated vaccine, 50% efficacy, but refrigeration for 2 doses. There's a CanSino, another Chinese vaccine. The efficacy is unknown, but can be stored at refrigeration temperatures and supposed to be 1 dose. That's a non-replicating viral vaccine. Just to remember, what we know right now that these vaccines, so the ones that are in the US that are authorized already is the Pfizer and Moderna vaccine. The next one is the Johnson and Johnson vaccine, and we may also see a couple of other vaccines, including the Oxford vaccine, in the next few months. But with all of these, we know that they are highly effective against preventing all disease, including severe disease. Um, Especially severe disease. So with severe disease, very little variation. But we don't know at this point whether they're preventing transmission. So whether they're preventing your ability to give the virus to others. We think that there will be, based on we know what we know about these kinds of vaccines and biological processes, there will be some reduction at least in transmission, but we don't know how much, and it may end up being imperfect reduction in transmission, your ability to give the virus or acquire the virus, even if you're not getting the disease. And so, therefore, the implications are that until it's not an infinite approach, until enough people are vaccinated themselves, the best approach is to, even after you vaccinate, you can do some additional activities. It's not, I'm of the belief that it's not that everything is the same as before, but still reduce your risk by wearing a mask in public, continuing to wash your hands, and maintaining 6 feet distance for other people's sake, so that you're not a vector, even if you yourself are protected. These studies are going on, and we are also looking into some of these questions. In our research group, once there's more data, what is the level of protection against transmission? There may be more changes in these recommendations, but for now, these are the recommendations. A few things. Pregnancy is increasingly a high risk, recognized as a high risk conditions if after you get SARS-CoV-2. It happens with other respiratory viruses like influenza. A lot of my own work has focused on that previously. And so we know that higher ICU admissions happen, invasive ventilation needs go up. with pregnancy if you get the virus when you're pregnant. So therefore, there is a need for, there are a lot of healthcare workers who are women in this country, and a lot of them could be, a substantial proportion can be pregnant this at a given time during the rollout of this vaccine program. So there are different recommendations, etc. But essentially, in the U.S. specifically, the idea is it should be made available. So CDC, American College of Obstetricians and Gynecologists, Society of Maternal Fetal Medicine, FDA, etc. So essentially, the details recommendations say that you make the vaccine available to pregnant women, but then they consult with their obstetric providers in terms of the risk versus benefit of their specific conditions. But it is very encouraging and permissive recommendation for pregnancy. In lactation, if people are lactating, it's even more permissive and encouraging recommendation. So people who are breastfeeding and so on and so forth. So that's the overview of the virus. I will pause here in the interest of time, and would be happy to take questions and respond to any queries.
Thank you, Dr. Omer. I'm going to unmute, give everyone the ability to unmute themselves so you can ask your questions verbally. You can also put them in the chat, raise your hand, and I can call on you. There is a question, Dr. Omer, from Joanna in the chat. She wants to know how the clinical participants were recruited so quickly and who recruited them.
That's a really good question. So there are different pathways. So a lot of these trials were coordinated by the National Institutes for Health. Others were, the Pfizer trial was coordinated by the company directly. Both of them eventually converged on the pathways, similar pathways. So they worked with academic medical centers mostly and other sort of medical providers to consent people, so to reach out to different individuals and communities, to consent them and follow up, and then sort of vaccinate them. Yale was one of the sites, and one of our colleagues was leading the Yale site for this, for the Pfizer vaccine. Another colleague of mine in Yildirim in pediatrics is in the process of setting a pediatric trial within the Yale system with NIH, etc. But the idea was that what NIH did and what Pfizer did, its own coordination, but a lot of these trials go through in the US through NIH, which is they utilized trial capacity that was built up for, let's say, HIV drug evaluation, all sorts of other networks. So they scaled up substantially. And so therefore, the model that I was talking about, a lot of sites with therefore reduced requirement per site of the number of people you need to enroll. That's how they enroll people. Initially, very initially, in the first couple of months, there was a concern that the initial set of recruitments were not as diverse as we wanted to be representative of our population in the US, because you want your results to be generalizable to everyone. But then there were efforts that were made. So by the end, both for the Moderna and the Pfizer trial, it was a pretty diverse pool of participants on which this vaccine was evaluated.
Thank you for that. Another question in the chat from Rick. Approximately how long does it take Pfizer, AstraZeneca, et cetera, to actually manufacture a vaccine dose in the factory production process from start to packaging?
That's a really good question. And I don't have, and we don't have that information directly because part of it is that it's their proprietary process, but it also depends on what stage of scale up you are. So earlier on, it takes more time. It takes a bit longer for a lot to be produced. Later, it takes less time, and you have scaled up manufacturing. I do know that there are efforts to nudge higher level of production, even amongst those who did the earlier authorized vaccines in the US. For example, the Defense Production Act is being used to say that, for example, Pfizer is a high priority entity, so that if they need any equipment to scale up production, any plants, if you want to expand their manufacturing plants, etc., they get precedence over any other supply in the US for sort of the kind of stuff they need to expand their production. So I don't have the direct answer to it. It is in sort of from weeks to months, but it depends on what stage of scaling up you are.
Does anyone like to ask a question? Otherwise, I can go to the chat. There are quite a few.
I can, well, I guess I'd like to know with the production of these vaccines, um, how far are they having to travel? You know, is production, say, in hubs, you know, like you have with the airports, so that you're fairly close to, you know, to different states, or is it in one place and it's traveling long, long distances?
So it is in a couple of places, um, but these places are close to transportation facilities because a lot of it is being delivered through our courier network in the U.S. So UPS, FedEx, etc. That is being utilized. So it is because it is more efficient to do things together rather than setting up smaller factories in a lot of places, and it is easier because they have to maintain quality for every single lot. And it is easier to do that if it's in one place or in a limited number of places. So, from the prediction efficiency, the total time to production to getting into the arms, it's more efficient to do a little bit more centralized. So, there's still, you know, multiple production facilities inside or outside the US. But a lot of these, the way that is handled is that it's not, a slow transportation process. The doses are being transported, uh, you know, uh, uh, more fast than just, the slow version of our transportation networks.
Thank you. Another chat question. Are efficacy rates known for only one dose of Pfizer or Moderna?
That's a really good question. They're emerging data. So these vaccines were not evaluated for two doses, and there is a genuine scientific. There was genuine scientific back and forth whether one dose is sufficient, etc. Because this vaccine worked really well in it had 95% efficacy, and there are initial data between the first and two doses. There are 2 to 4 weeks between Pfizer and Moderna. Pfizer is 3 week difference between the first and second dose. Moderna is 4 weeks difference. So if you look at the data, you know that the vaccine starts protecting even after the first dose. How much it is, there's some sort of range in this, and so there was some thinking. Some people sort of raised this issue, that maybe we should just stick to one dose and increase the supply quickly of just giving one dose to everyone. Uh, then the scientific consensus that experts that independently advise the FDA and similarly at the WHO side was that, look, because of the fact that we have variants coming, because of the fact that we are vaccinating diverse populations in terms of age and other background, in terms of comorbidity, etc., like the other conditions they have, let's stick to what we have and what we have direct data for and for which there was licensure, and then do studies in parallel to see if one dose it may be sufficient, but not jump to the policy before carefully studying that question. And so therefore, the consensus is that the recommendation remains of two doses.
Great. And I'll squeeze another chat question before we go back to the gallery. What are the raw materials used in the vaccines, and is there any danger of running out of them?
That's a really good question. So there's a lot of other things that are used. The two mRNA vaccines don't use an adjuvant based on their technology. That's what they're one of their advantages. But there's several other things, the kinds of lipids, sort of micro level fats, etc., that are used as part of that whole package in the vial. They are always supply, you have to keep an eye on this, but also for glass that is needed for look, we globally, and it's a global market, we are looking to vaccinate approximately 2 billion people within the next year and a half, or at least 1 billion people within the next year. And so obviously you have to look at the supply chain. And so the glass that I was talking about that can sustain ultra cold chain level temperature, et cetera, the kinds of syringes that draw, there's a kind of syringe that draws one extra dose that's what we call dead space, et cetera, and so on and so forth. All of these things are considerations of the supply chain. Uh, so far, we haven't faced bottlenecks on the supply chain at that level. There are a little bit of things that could have been sped up for on the manufacturing side. So the manufacturing, not what is in individual dose, but what we need to sort of expand your manufacturing. And so therefore, hence the focus on the Defense Production Act recently to ease up some of those constraints that even if we are able to save a few weeks there in terms of companies having access to that kind of equipment. But overall, in terms of individual supplies, it wasn't that governments and companies didn't have to troubleshoot, but with that troubleshooting, things are okay. That's not sort of the current bottleneck right away.
Reese McLeod has a question.
Good morning. Thank you, Karen. Good morning, Dr. Omer. I have parents who are 65 and older. My mom is 72, my dad is 75. And of course, they're not tech savvy. So I've been the person that has been going online for them. I've been calling. And within the first day of the 65 and older, because my dad is only 64, my mom is 72, they were eligible on Thursday. And calling, I was online for hours with no resolution, to the point where I can only register them for VAMS, I believe it is.
Yeah,
but there was no appointments, where I would get sites saying there were available appointments. And then when I would get to the site, there were no appointments available. What is being done to assist in ensuring that the 65 and older are getting the vaccination and finding locations that are close to their home and not miles away? Because I noticed that there were places like 20 miles, 15, 20 miles away from my parents' location in Hamden, which they're older, and especially during this winter season, it's hard to travel.
You're absolutely right. And this is a big bottleneck. And one of the things that a lot of us have been, I'm on record in June, actually starting May, but specifically increasingly vocal in June of 2020, jumping up and down that this is an unprecedented vaccination effort that we're going to undertake, especially for adult vaccination, especially for the elderly amongst us. And so we needed to prepare for this. And this is what exactly preparation means, that the VAMS system is a reasonable starting point, but it needs to be more user-friendly, et cetera, less onerous, but then still backed up by volunteers that reach out to our elderly population or work with them. You shouldn't be, someone's life and death should not depend on how much time their grandchild has to take them through the system. We should do better than that. So that's a blind spot, to be very honest, in this. Some of it will be eased up as people get used to it a little bit more. Some of it will be when there are more volunteers or sort of paid workforce added to help navigate people. But right away, it remains a bottleneck. Just to say that Connecticut has done a better job than many other states. But the problem is that everyone is in that same boat that we, so the example I give that when the preparation was happening, the Operation Warp Speed, for example, that you may have heard of, did a really good job of sort of finding candidates, investing federal money in speeding up the development of it. They really did do a good job of that. But what they didn't do is to prepare to deliver these vaccines through a fragmented healthcare system. And that continues to worry us. So they built a fancy, shiny plane, but forgot to, forgot to build a runway to land it. So So they're trying to land a 777 on Tweed, if you will, and so that kind of a mismatch between uh, the technology and the health delivery is what we are seeing. So it is unfortunate. There's steps being taken, and I would think eventually things will pick up. And for example, if we go to a phase, the seniors who are left out in this phase, if we go to a phase where there's enough vaccines that everyone is getting it, so there's less requirements for appointments, etc., through mass vaccination. If we ever get to a phase where there's walk-in appointments, etc., so things ease up a little bit. But you want to vaccinate uh, older age individuals in their 60s, um, especially 65 and above, earlier on now. And this is what we are paying for in terms of lack of preparedness on the delivery side while doing a good job on the development side. Sorry,
Okay.
I don't have a more satisfying answer, but it is frustrating for a lot of us.
Thank you.
Thank you. I have a few questions before we go back to the gallery. How long is the Moderna vaccine viable out of the freezer?
So yeah, so it's when you open the vial. So the different steps there. During a day, it's a different requirement. When you open the vial, it's 6 hours. So some folks may have seen that story in New York Times, where a physician in Houston tried to just basically ran around vaccinating people when the dose was missing. There is consensus. I've talked to other colleagues that said there is consensus that he did the absolute right thing. And we should have a waiting list system, but especially initially we didn't. Connecticut is, again, better than other states. There's a specific requirement how to maintain a waiting list now, and but even in initial stages, even Connecticut didn't have clear guidance, because we were sort of setting up, like the state was setting up um, the program, but there is clear guidance, and I think he did the right thing, if you're following that story, by going through the risk groups, asking the first responders, including police that were there, offering to them, and then even when he went around and vaccinated people, he went to the highest risk groups, etc. He didn't, vaccinate the pizza guy he found on the road. But even then, I would say I would go to the extent, if you don't find high priority, these are precious doses. We should not be wasting them. But that's the practical side of it. Once you open it, it's 6 hours.
Thank you. Is there a difference in vaccine effects for those who get the virus versus those who did not get it?
We're looking into that. We don't think, based on other viruses, that there will be a difference in safety, like long-term safety, between those who have had the virus. And my group is actually looking at that specific question as well. Um, uh, but in terms of long-term safety, but we do expect that those who've had the virus may have transient symptoms in higher proportion, not necessarily more severe symptoms, but a higher proportion of them may have, the symptoms that look like the second dose. Even after the first dose, the proportion doesn't seem to go up when you get to their own second dose. But it does seem to have that, your first dose feels like your second dose in some of these people. And these are early data. So, watch this space. We will have more data. But in terms of what we know about the biology, et cetera, of viruses and vaccines, we don't expect to have severe side effects to be different between the two. It's these kinds of things, fever, headache, fatigue kind of things that I'm talking about.
Thank you. Now, I see 3 or 4 more questions, but it is a couple minutes past 9. Do you have a few more minutes, Dr. Omer? Or would
Let
you
me
like?
actually look at my calendar. Actually, no, sorry. I need to go for a CDC meeting
Okay.
that I can't sort of skip. I'm on Twitter. If y'all want to follow me, at Saad Omer 3. I don't know who the first two are. I kind of have a slightly different name, so I was a bit surprised. So it's Saad, Saad, and last name Omer 3. You can follow me. I often talk about these vaccine and COVID-19 related issues, and some dad jokes, but that's a
Well, thank you very much for joining us this morning. This is an excellent talk. I really appreciate your time. I encourage everyone to follow Dr. Omer and reach out to him with your questions or reactions to his jokes. So thank you. Thank you
Thanks.
very much.
Very
Have
nice
a good
talking
day,
to everybody.
everyone.
Bye.
Bye-bye.